Okay, great. I switched gears. So maybe having the afternoon session early. So I'm going to talk about the driving forces for the cell fate decision making. And we're going to show that the landscape is not just a metaphor, it's physical. It can be measured, it can be quantified, and it is useful. But the landscape itself is not enough to characterize the dynamics. You need another component, the driving force, which I call that flux. So it's a dual driving force, which I'm going to talk about. So let's, since this group doesn't need an introduction on this, so let's jump directly to this. The x dot equal to ff is the driving force for the dynamics. And the typical way of analyzing this is just find a fixed point and doing the local analysis, stability analysis around those fixed points. Now, this kind of analysis does give you the local information, but what we care about in the cell fate decision-making context is not just the local stability, is how you go from one state locally stable to another local stable point, which is the cell fate decision-making. And for that, you need the global information. And such global information is not present in the typical local stability analysis. So we need something global, but this global thing has already been rooted in the physics and chemistry for the closed system and the equilibrium system. And equilibrium system, we know two things. One is the interaction, and the equilibrium probability. Equilibrium probability is just the weight of the states. And once you know the weight of the states everywhere in the state space, then you can connect the two states right together. And because you know the in between what the bridges are, what is the weight, what the weights are. So you connect them too. And then you form these valleys and the mountains that gives you the difficulty, how you go from one to the state to the other. So this is the global connection. Now, the local connection comes in because the gradient of this landscape it determines what you are going to do next. What are you going to go next? Okay. So this idea has been applied to many different biological systems. And ranging from the dynamics down below the dynamics of the biomolecules to differentiation, development, and evolution, actually context. I would say that for the biomolecular aspect, it's spontaneous, so we know it's very similar to equilibrium dynamics. But for the differentiation and evolution, I would say it's although when the pioneers view that as a gradient case, it's actually not. But if we go beyond the protein, the biomolecular level, going one level up to the cell, and you can think about the cell as many chemical reaction soup and chemical reaction networks. One can often find write down the equation like this, you don't have a gradient. The force is typically not a gradient. And then you can say, okay, how do we define the landscape per se? So I'm going to skip some of the details of the math, but just give you the answer that although the force cannot be written as a pure gradient, and so that it looks like the landscape cannot be defined, but it can be defined by splitting up the forces into two. And the one force is the landscape-like, is a gradient-like force, which is a gradient of this landscape. This landscape, instead of in the equilibrium situations where it's directly related to the equilibrium probability, now it's linked to the steady state probability distribution. On the other hand, so this is the equilibrium-like or gradient-like force. On the other hand, the other part of the force has two important ingredients. One is it represents how much, this is a open system. For the closed system, you have a gradient force, but for the open system, you have matter input, you have input from the energy or information, and this net flux in, that's what it represents. as an open system. And once you have that not equal to zero, and you have this force, but not only that, this force has a rotational nature. So two natures representing the input, net input, and therefore break the detail balance becomes the measure of non-equilibrium, and the other is rotational like. So what does this mean? This means that you have a landscape, and in addition, instead of going down, slide down like gradient force, you have a spiraling force going like this. And what's the consequence of that? That typically you have like a two basins of traction, two states. You go from one state to the other. Instead of going directly as we expect, going through the saddle point where you use saddle point to determine the barrier height and determines the rate from going from one place to the other. Because of the presence of this rotational force, you won't typically go through the transition state here, the saddle point here. And so, and not only that, going forward and backwards are not going to be the same as the past. And therefore, when you want to determine the rate, self-fit decision rate from one to the other, you have to determine what's the optimal point on the path rather than the saddle point. And so this is what's important. Not only you need the landscape per se, you also need to consider the rotational force component. Flux comes in. Yeah. Go ahead.
Optimal point that you mentioned,
yeah.
Is that like related to the transition state in the sense that you have 50% chance of going forward or backward? Or?
Yeah. And also the thermodynamic law is going to be generalized because the entropy production rate is not just for the relaxation process, because at the steady state you still need energy. So this is the housekeeping related to this flux. This is the extra heat you need to maintain at a steady state, because if we don't eat and drink for 10 days, we die. I mean, that's you need some energy to maintain your homeostasis state. And the other thing I want to mention, because yesterday Mike mentioned about this, is the fluctuation dissipation relationship is going to be changed, because the response is not just equal to the fluctuation in the equilibrium. It's the response is equal to the fluctuation in the steady state plus a correlation of the observable with this flux force. Okay. And so let's have an example of this. This is a pre-single cell sequencing times. So we have this gene network, or mostly like the protein networks, that determines the cell cycle. I mean, cell cycle is a fundamental function of the cell, and through this different stages of the cell. So let's see what's the usefulness of splitting up to two forces. So you have the typical textbook of this kind of cell cycle going from G0, G1, and going through the checkpoints and SG2, and then checkpoints going to the M checkpoints, and then split up. And in the orbit is sort of running a circle, and then they split. This is a cycle. And so this is the vertical axis of the landscape, and is the landscape, and those are the two gene expression on the horizontal axis. And what you can see is the landscape. This is the minus log of the steady state probability distribution when you look at the dynamics. It's actually an irregular Mexican head shape. And so where you are, anywhere you are here, on the state space, you get attracted by this gradient. So down to this ring, the closed valley ring. But once you are on this ring, and you will likely be more likely to stay here because this is the lower potential point. Actually, what happened is that if you just have the gradient force, you will be there. You won't move, and so that's where you need the flux force, the rotational force to rescue you, because it is the rotational force once you are in this valley to drag this to go around, have a coherent oscillation. And also you notice that along this line, there's a local basin of attraction, and going through this local bumps, which is characterize the checkpoints over here. And so what's sort of the usefulness of this is twofold. One is because the landscape that this basin, the island height is global, and the flux is global, and that they can be used as some marker to characterize how they change will affect the function of the cell cycle. For example, if the global flux is big, and then of course it's going to be faster, and cell cycle will be faster, and we know that cancer cell is much faster than the normal cell cycle. And the other thing is the quality of the cell cycle is determined by the height of the barrier. And the height of because if this guy is low, you can go directly to here. You don't realize the direction comes from here or from the other direction. So you lose the coherence. So you need this to be high, and you need the global flux to be high. And using this to do the global sensitivity analysis, you will find out which components of the gene networks are crucial for determining this quality of the cell cycle. So this is the one aspect which is good. The other aspect which is very interesting is the following. This thing depends on the environment. The landscape depends on the environment. If you feed them with no nutrition supply, then it turns out the landscape looks like a single attractor, which is the G0 situated G0, G1 state. When you increase a little bit, then it keeps on going, and you increase a little bit more, and then you start to have this kind of a bistable G1 G2 state. And if you keep on going, then you start to have this kind of oscillatory behavior. So the landscape actually changes according to the nutrition supply. So this also means that in order to have a cell division, you really need to pump in enough energy to be able to have a cell living, a living cell. Yeah, go ahead.
Just curious, like, what does the flux represent, like biologically speaking?
Biologically speaking, it's basically well, I mean, I'll give you this. It represents strongly correlated to the energy supply. So you have ATP hydrolysis, then you have this flux. If you don't have sort of any pumping process, this term is zero. Yeah. Okay. So another one, that famous one, we can't escape from that, is the Waddington landscape. I want to show how you actually quantify this Waddington landscape by a simple circuit, mutual repression, self-activation. And then you can work out the dynamics, except one thing is that we know that from the experimental studies, especially the Sui Huang studies, that the regulation changes according to during the developmental course. And so this regulation actually changes either A or B in different contexts. So you can think about this is gene circuit, but with some regulation changes during the development. So this axis is the gene state axis. This is the landscape. And during the development, this is where the differentiation changes during the development. This is the developmental direction. So this is the landscape at a certain stage of the development. So Waddington landscape should really be viewed as a landscape, dynamical landscape along the development. And it can be quantified. This can be quantified. And what's interesting here is to show that the paths of differentiation and reprogramming are different. And this is a reflection where you have this rotational component in it. And even furthermore, to show you that both landscape and the flux are important, is when you look at the landscape, this is still another version of this landscape. You see that if you plot the flux distribution or sort of the flux value along this developmental direction, you find that near the bifurcation, actually the flux becomes big. And there's a reason for that, why it doesn't prefer to be in the stem cell state. It wants to bifurcate. is because the gradient force always try to converge. It wants to comfortably sitting in the local space. But on the other hand, flux is rotational, so it wants to spread out. So it's the competition from one to the other, at least that eventually to split up, to bifurcate for this non-equilibrium bifurcation. So we tried another bioinformatics-based networks and do this global stability analysis and find out that the solid lines, the octal shapes, are the key genes and key regulations. And many of them are familiar friends that people have known about, crucial for differentiation and the reprogramming. But one stands out, which is not known before, is PBS1. And the experimental friend look at this and further experimentally and identify that has a biological function enhancing the IPS generation, and it's a therapeutic target for the.
That network that you showed constructed.
This is a bioinformatics constructed.
From what?
From the bioinformatics data, and this is this is not this is before the single cell sequencing.
Sure, but how do people know the arrows? So there's some bulk RNA-seq data that they use to get these arrows. Like, what is the?
Yeah, there are some studies then showing the errors. Yeah.
So, my pushback would be like, it's as good as random. I don't think many of these arrows mean much if they have not been causally tested.
I think from transfer or transcriptional regulation,
have an individual knockout to get this network. So
no, it's known from the binding sites.
Oh, but it's still inference, though, right?
It's a transcription factor binding site, and some are backed up by experiments.
Okay.
But yeah,
I just don't think so many arrows can be mapped confidently in a network.
These are well-studied transcription factors, so I don't know.
That's fine.
Yeah, I see your concern.
Yeah, just for big networks, it's a little tricky to know.
But they're all well known.
Yeah. Okay, sounds good.
Okay, so this is the therapeutic target, and it has the sort of a function of the synesis and apoptosis target, and so far, we talk about how to the examples of how to quantify that. But what about the experimental? Can we measure it? So we try a few different things in collaboration with the experimental group. One is the typical classical ones, the toggle switch. So by doing the temperature sensitive mutations, they are able to look at the real-time trace of this and collect the statistics, and you can see this is the landscape, the distribution. And we can see there are, instead of 2 states, we actually see 4 states. And you can, through the hidden Markov chain simulations, you can get the rates of going from one state to the other. The other one is, how about flux? And we collaborated with the experimental group. They consider single molecule enzymatic reactions, and the one enzyme catalyzed 2 substrates instead of 1, and with the conformation changes. And what they have seen is non-Michaelis-Menten behavior rather than the typical Michaelis-Menten behavior. And this difference gives you sort of the flux, and you can deduce from that. And the furthermore is the one we look at with the experimental group is the pheromone response, and this one is related to the shape changes or the morphology shape changes, and then we can see that at a certain dose, and the morphology has great changes for the populations, and this is also accompanied with the signature of the flux force increased tremendously. So now I just go to the sort of the single cell RNA-seq studies. I don't have to introduce this. And with the RNA velocity and the force field, and we can get to here, and then we can ask the question: can we decompose the force. And that's what we did. We decomposed the force. And for cell cycle and cell sequencing data, we found that this is the landscape we came up about, and this is the flux flow, and this is the landscape. And you can clearly see the G0, G1 as G2, and all these phases going around. You can see the going around the loop. This is the Mexican head landscape. You need both force to drive the coherent cell cycle. And so you can also study the effect of this flux force and the associated dissipations and how you suppress the phase diffusion and increase the quality of the cell cycle. And we, of course, go on and look at the differentiation. Then by splitting up the force, we can see this is the progenitor cells differentiated to several branches. And you can get the optimal path going from one to the other. And the one thing we want to notice that these paths, if you look at, maybe this is not a very good drawing, but if you notice that the path, the progenitor going from one state to the other, they don't typically go through the settle point. And if they don't go through the settle point, we use the action to measure the optimal point where they are supposed to be the transition state. It's path dependent. This is where we identify the transition state with the actions. And then after that, we ask the question, at this point, ensemble, transition state ensemble, we ask the question, are some of the genes expression profiles, are they close to the end? If they are close to the end, means that at the transition state, they are already formed. And if they are not far away from the end, and it means that they are not formed. So the ones that are already formed and the same as the end, that's the nucleation seed. We call the nucleation seeds. And those are the ones that the key ones are the pioneer genes, and the rest of them are followers. That's how we physically use to see this. And so that's the sort of procedure, using the actions to get the transition state and the pioneer genes. So I'm out of time. Just here's the summary. So we identify the global driving forces, the landscape and the flux, and both are needed. And we pointed out in the cell cycle, it's absolutely necessary to have two forces. And even for the landscape, for the decision making, for the differentiation and reprogramming, it's useful because it's essential for the bifurcation when it happens. And finally, we can see using the low throughput single cell synthetic biology experiment, we can quantitatively measure them and see their effects. And finally, single cell transcriptomics, we can do cell cycle differentiation. We can use that to identify nucleation hotspots for pioneer genes. Okay, thank you very much for your attention. Go ahead.
Thank you for that nice talk. I really like how you can sort of, isolate how the genes are changing over the transition path and pick up like non-monotonic relationships. But it seems like it's very dependent on like the dimension of your system and probably works best in like 2 dimensions. So I'm wondering what assumptions go into, using, so for example, a 2-dimensional UMAP to do this sort of analysis.
Yeah, I think this has always been an important question of how you identify the reaction coordinate or the other parameter of the system. And I think so far there are several methods like a UMAP or a PCA, all of these things, and they are not exactly the coordinate, the reaction coordinate, or the other parameters. I will say we haven't really found the reaction coordinate and the other parameters, but this is a challenging question that I think the research direction should go. It's a big challenge. and it's a general issue of how you go from complex system to compressed dimensions and reduce to the point where you have the best reaction coordinate to follow. Yeah.
Thank you.
Yeah, go.
Thanks. Yeah, very interesting. We have different approaches, but in your case, you explain a branching being driven by the flux,
right?
That overcomes the gradient.
Yeah.
But that doesn't necessarily follow the geometry of bifurcation theory. Is that correct? And where does the flux come from? I think it's Adam's question. So you need some actuator to change the flux, right?
No, this is because the parameters keeps on changing. Because the Waddington landscape, one direction is the parameter. The parameters is actually
that affects the flux.
Yeah, that affects the flux. Yeah. Different parameters like your self-activation or mutual repression. When that changes, the flux changes, and only when that changes, the bifurcation structure. You start from the same.
Time for going to have anything that's oscillatory, you're going to have to change the flux, right? That's basically you need the flux. So basically, otherwise, regular landscape doesn't work if you have oscillations, right? So, but fast oscillation may be many other things. Can be ATP, can be but
But like, what if it's just not like? Obviously, it's a very different force, but it was just landscape plus like gradient plus noise, stochastic fluctuations.
Doesn't give us.
It doesn't give you limit cycle. Limit cycle cannot exist with this.
Definitely see it in the cell cycle. I mean, other people showed it as a set of attractors rather than a limit cycle.
Well, clock.
Can I make a trivial mathematical remark?
Yeah.
Really trivial. If you have an attracting periodic orbit, you then have, literally that. The attracting periodic orbit will have an angular frequency as a function of where you are in the angle. And of course, that will give rise to what the time average distribution is on where the states are. If you're slow, you'll build up probability there. And that's it. There's, a periodic orbit's a periodic orbit. Nothing more or less. It's a periodic orbit.
Can I comment on that? Yeah, we saw that too, but then we did a lot of cell cycle measurements.
Yeah.
And you mentioned cancer cells divide fast. They actually don't divide faster. They spend more time in the limit cycle. So it's hard to change. It's very hard to change the period of a cell cycle.
I don't know what divide means unless you're going around the cell cycle and it's a periodic system. I mean, if they're doing something else and they're blocking in G1, in G1S or G2 prior to division, they're doing something else. So it's not a simple limit cycle. You could ask how the limit cycle is lost in a mathematical sense, i.e. there could be a SNC or some other, which is interesting as to then where they go. But it's all simple dynamics. Physics.
Yeah, there is an interesting aspect because it's different from the clock because the clock has the time reversibility, and this one is not. It's a limit cycle. It goes only in one direction, but the clock, like usually the pendulum, and the pendulum is different because the pendulum is oscillatory, but it has a reversible flux, and so it's not a one direction. This is a one directional flow. The limit cycle is one directional flow.
Clearly, life is non-equilibrium. We grant that. If it's equilibrium, it can be very interesting.
Yeah.